Hello students, a very good afternoon. So today we are going to talk about biomolecules. So before we start the session, as usual, we are going to have a technical check. So I'm going to launch a technical poll. Please let me know if my audio and video is clear. Okay. So here's a poll on the screen. Please confirm if the audio and video is clear. And the YouTube students, you can just type in on the chat box. Okay. So I need a confirmation on audio and video. So anyways, we are approaching 1 minute time. So if you have any technical problems anytime, please put it in the chat box and we'll start our session today. Okay. Fine. So we are going to talk about biomolecules, one of the most interesting concepts that we can come across in biology. So it is an interdisciplinary subject, right? We are going to talk about chemistry as well as biology, right? And we understand the importance of chemistry in biology for the first time. And having a basic understanding of biomolecules will give us an ability to understand so many of the biochemical cycles that happens in plants and animals, right? So you might have studied in your 11th standard about glycolysis or aerobic respiration or anaerobic respiration, right? Or some of the cycles that happens like Krebs cycles, right? Electron transport chain, and all the enzymatic reactions that you come across in the digestion. So if you see most of our living organisms, we all have principally the same type of elements present in our body. So we have hydrogen, oxygen, carbon, nitrogen, calcium, magnesium, zinc, iron, right? So if you just imagine, it's the same kind of elements that is even present in our earth crust, correct, right? So what is the difference between a bacteria and a human being? If I ask you a question, what would you tell? You tell that we are unicellular, they're unicellular, we are multicellular, right? We are more complex, they're less complex. So they cause disease, and we don't cause disease. We are intelligent, and they are less intelligent organisms, something like this. But if I take a bacterial cell, and if I take a human tissue and analyze, okay, so you might have remembered one of the key experiments that we conducted in the +1. What did we do? We take any living tissue of plant or animal matter, we subject it to trichloroacetic acid, and we analyze two different types of pools. One is acid soluble pool and acid insoluble pool. So we understood that we both had similar type of micromolecules and macromolecules, right? Irrespective of what origin we are and what complexities we have, we all have the same kind of biomolecules, right? Such as you can talk about proteins, you can talk about lipids, you can talk about nucleic acids or carbohydrates, or even the composition of the enzymes, right? So let us go and understand the key aspects of this chapter. So before we start this chapter, as usual, we have a test, right? So the first question of the day is here. The two functional groups, characteristics of sugar are hydroxyl and methyl, carboxyl and methyl, carbonyl and phosphate, carbonyl and hydroxyl. So here are your options. So let me launch a poll. I want you all to participate. Okay. Even though if your answers are wrong, it's okay. But I want you all to answer your opinions. Okay. What are the functional groups of sugars? So our options are hydroxyl and methyl, carbonyl and methyl, carbonyl and phosphate, carbonyl and hydroxyl. Okay. So let me know. Which of the following are the functional group or functional characteristic groups of sugars? So that's your first question. Make it fast, kids. You already studied in your +1. So it should be quite easy for you, right? It should be quite easy for you. So let me see what kind of answers we get. So our time is up. I'm going to end the poll. So let's see what's the right option. See, the sugars can be classified into two major groups. One is aldoses and ketoses, right? If you see aldoses or ketoses, we have two functional groups. One is carbonyl groups, other one is hydroxyl group. Okay, so you can see in the next picture what we can say. So you can see here, if you talk about aldehydes, okay, if you talk about aldehyde group, we have carbonyl group, correct, right? Sorry, hydroxyl groups. If you see in ketones, we have, okay, we have hydroxyl group, something like this, right? So our option is the fourth option, that is carbonyl and hydroxyl group. We'll learn about this in detail in the coming slides. Okay, no problem. So here is your second question. The major role of minor elements inside living organisms is to act as cofactors of enzymes, building blocks of important amino acids, constituent of hormones, okay, then a binder of cell structure. So again, I'm going to launch a poll for 1 minute. Make it fast, people. Okay. So some of the minor elements which are inside the living organisms, they act as what? That is our question. And YouTube students, please try to answer in the chat box. We have our second question, students. The, the major role of minor elements inside our living organisms act as what? Cofactors of enzymes, building blocks of important amino acids, constituent of hormones, or binder of cell structure. This is our question. Make it fast. We can learn about this in detail. Okay. So it's almost 55 seconds. We have to end the time because we only get 1 minute in the exams, right? So I'm going to end the poll. So let's see what's our right option. Okay. So when we talk about minor elements, so minor elements can be something like zinc, NAD, okay, or it can be like magnesium, it can be like iron. So these are called as cofactors. Okay. So they usually act as cofactors for enzymes. So we know about cofactors, right? So an enzyme which is produced, they are in its inactive form. So with the association of the cofactors, okay, the enzymes becomes active enzyme. So those are cofactors. For examples of cofactors and the list of cofactors will follow. Okay, next one. So today we are going to cover some of the important portions. So some of them are, we have to talk about what are carbohydrates, what are proteins, lipids, and enzymes. So which are basically macromolecules. Okay, so let's go ahead. When we talk about biomolecules, so what did we do? I was talking about initially what we can do with a living tissue. So I told you, if I take the living tissue, whether it is plants or animals, and subject it to, trichloroacetic acid. So why do we use trichloroacetic acid? If I take my tissues, as one cell is attached to another cell, right? So this gumming, what we call it as, so it has to become free so that the cells can come out of the cementing that is present in the tissues. Okay, that is what we call it as ground matrix. So once we relieve the cells out of the ground matrix and then we grind the cells properly, we get two types of portions in that, filtrate. Like we have the soup, we filter the soup and we get the filtrate. So once we analyze this filtrate, what we can say is, one, we can see it as an acid soluble pool, and other one is an acid insoluble pool. Whichever is insoluble becomes macromolecules, whichever is soluble becomes micromolecules. So why do we call it as macromolecules and micromolecules? Macro means big, right? Macro lenses, right? So when we talk about micro means small amounts. So we can determine the, what type of molecules based on their molecular weight. So if I talk about micromolecules, they should have their mass, which is around 18 to 800 daltons. Okay, then we call them as micromolecules. So they are present, as I said, they are present in acid soluble pool. Remember this, they are present in acid soluble pool. So where are they present in our cells? in all living organisms, we can come across this acid soluble pool or biomicromolecules, usually in the cytoplasm. Understood? So can we list some of them? Definitely. So here is the examples for micromolecules. Amino acids, lipids, sugar, and nitrogenous bases are the examples of biomicromolecules. Okay. So when it comes to macromolecules, if they have the mass more than 1,000 Daltons, okay, so they are very heavy molecules, such as proteins, polysaccharides, and nucleic acids. They are the examples of macromolecules. Their mass is so high they can reach up to 10,000 Daltons. Okay, so where are the macromolecules present in our in a fluid that we in our solution that we extracted? They're present usually in the acid insoluble fraction of that thing that we extracted. Okay, so they are usually structural and functional particles. For example, you can talk about the composition of the cytoplasm, the composition of organelles, the cell membrane, okay, and even the energy molecules like glucose, or you can talk about some of the cellulose. Okay, so they all come under macromolecules. Nucleic acids like DNA and RNA, they're also part of biomacromolecules. Okay, people. So when we talk about some of the micromolecules, first we go and understand carbohydrates so that we can split these micromolecules and macromolecules accordingly. So carbohydrates are one of the important factors for our cells and all the living organisms. The reason is very simple. So if we talk about how do you gain energy, the first thing that you're going to say is we gain energy through glucose, right? So we eat food, and the food is broken down into simpler form of carbohydrates that we call it as glucose. Correct, right? So if we talk about the composition of the cell wall, or if we talk about any of this, okay, cell wall or the storage of food in the plant cells like sucrose, so all of them, they come under carbohydrates, right? So now carbohydrates can be of two types. Basically, they are either polyhydroxyaldehydes or they are polyhydroxy ketones based on the presence of their functional group. So if a structure has CHO group, then we call it as aldehydes. Example is ribose and glucose. If the structure has CO as the functional group, then we call it as ketosis, or polyhydroxyketone, which is ribulose, and we have fructose. Okay. So based on this, we have monosaccharides, oligosaccharides, and then we have polysaccharides. In carbohydrates, there are two important categories again. So I will give you an experiment. I'll give a reagent called as Felling's reagent. And this is a kind of a blue color reagent. So if I give this Felling's reagent and ask you to treat our monosaccharides, disaccharides, and polysaccharides accordingly, what you can see is, you can see that these monosaccharides and disaccharides, they change the color. So the color change happens because they are reduced with the action of Felling's reagent. So whichever the sugar that acts with a Felling's reagent and reduce, these are called as reducing sugars. But in disaccharides, we have sucrose, which is a non-reducing sugar. Only disaccharide that do not reduce is called as a sucrose. Okay, you have to remember this example. When it comes to polysaccharides, we have I'm sorry, students, just give me a second. Okay. We have some network issue. Okay. Yeah. For a better way of understanding, let's go back again and do this. Okay. Share. So we were talking about different types of sugars. Let's continue from there. Okay. Students, is my voice audible since you're present now? Can I get a confirmation? Quick confirmation. Please give me a thumbs up or put a message in the chat box. So I'll be continuing my class. Please let me know, okay? So if you have any problems. So as I said, we call this glucose, fructose, and galactose as monosaccharides because they have the same monomers repeating up to 4 to 9 or 4 to 8 repetitions. So these are called as monosaccharides containing a same or single sugar unit, okay? When it comes to disaccharides, we have 2 sugar molecules, okay? For example, if you split lactose, we have glucose and galactose, okay? We have 2 different types of sugar units repeating, okay? One second. Thank you. Thank you very much for the confirmation. All right. Similarly, when we go for oligosaccharides, we have more than 2 to 10. For example, we have raffinose, okay, which is one of the oligosaccharides. But when it comes to polysaccharides, we have homopolysaccharides and heteropolysaccharides. Take, for example, glucose or cellulose. Cellulose is an example for homopolysaccharides. It is a repetition of glucose for several times. Okay. So we can write it as C5, H10 O5 to the power n. So this is the general formula, or this is the formula that we can represent for cellulose. So we have the same glucose monomers that is repeated for n number of times. So that will give us polysaccharides, right? If we have more than one sugar, we get heteropolysaccharides. Okay, so this is about polysaccharides. So as I said, we can divide our monosaccharides into 2 important groups. One is aldoses, other one is ketosis. Okay. So we have the examples for aldoses, that is glyceraldehyde is one of the examples, ribose, okay, xylose, glucose, mannose, okay, and heptose. These are the examples for your, aldoses. Whereas ketosis, you have dihydroxyacetone, okay, glucose, okay, fructose. So these are the main examples that we have to look into while we are understanding aldoses or ketosis, okay. So under this aldoses and ketosis, we have trioses, tetroses, pentoses, hexoses, and heptoses. Now, how do we determine this? If we tell trioses, they are the repetition of 3 sugar units. If we tell tetroses, they are the repetition of 4, penta 5, hexa 6, and 7, like this. Okay. So when it comes to oligosaccharides, disaccharides, trisaccharides, and tetrasaccharides, based on different types of sugar units. You take about disaccharides. All this contains only 2 types of sugar, that is sucrose, maltose, and lactose. Whereas trisaccharides, we have raffinose and we have maltose, right? Similarly, we have trisaccharides, that is stachyose. Okay, this is another example for oligosaccharides. So the classification is very, very important, people. Why? Because in our examinations, usually they point at, glyceraldehyde comes, whether it is aldehyde or it is a ketone, whether it is a triose or whether it is a pentose. So this slide can really help you to, just reduce your burden in searching for examples when the question comes. All right, fine. So as I said, we have polysaccharides, and polysaccharides can be divided into homopolysaccharides and heteropolysaccharides. Examples for homopolysaccharides are, as I mentioned, we have starch, we have cellulose, we have glycogen, pectin, dextrin. So all of this have the same sugar repeated for n number of times. So that is why we call it as homopolysaccharides. When it comes to heteropolysaccharides, we have hyaluronic acid. Okay, so hyaluronic acid has a very funny story. Usually we use this hyaluronic acid for our skin treatment. Okay, so whenever you have the wrinkles or whenever you want your skin to be young, we use hyaluronic acid, right? So we have heparin. Heparin is also called as anticoagulant. If we don't want our blood to coagulate, okay, our body produces heparin to keep our blood thin so that it can circulate better. Okay, heparin and hyaluronic acid are examples for heteropolysaccharides. Okay, so sugar combines with other prosthetic groups. Okay, I'll just mention sugar can be combining with other prosthetic groups. When it combines with other prosthetic group, it will give rise to derivatives of sugar. For For example, we have amino sugars. So if an amino acid is attached to a sugar group, then we have amino sugars. Examples are glucosamine and galactosamine. Glucose is a sugar, okay? And then we have an amine group attached to it. That is why we call it as glucosamine. Galactose is a sugar, and then we have an attachment of a prosthetic group like amine. It is called as galactosamine. Okay. Then we have deoxy sugars. Example, deoxyribose. Okay. Then we have fructose. We have okay. All right, people. Yeah, we have deoxy sugars. In deoxy sugars, either we have deoxyribose or fructose. Okay. So next we have polysugar alcohols. So we have sugar group attached to an all group, or like OH groups. So that is, for example, mannitol is an example for derivatives of sugar and an alcohol. We have sugar acids. Okay, we have galacturonic acid. Okay, and ascorbic acid. Okay, ascorbic acid. Okay, ascorbic acid. So these are the examples of acids. And there are other derivatives. Okay, for example, you might come across glucolipids or glycolipids or glycoproteins. You see, glycolipids, glycoproteins. So these are very evident that okay, these are the derivatives of sugar. Okay. Next we have, see here, methyl cellulose. Cellulose is a sugar. Methyl is a prosthetic group. We have carboxymethyl cellulose. Carboxymethyl is a prosthetic group, and we have cellulose as a sugar. Okay, so these are all derivatives. Please remember the examples. Okay, go through the examples. So let's go for proteins. So what are the importance of proteins? So if I ask you, so why do we need proteins? Some of you will tell, so we need proteins for, building tissues. We need proteins for construction of cell. So we need proteins for signaling. Okay, so many answers might come with respect to proteins. Okay, and the reason for that is because protein functions, okay, protein acts on almost 98% of all the biological functions. Okay, give me a second. 98% of all the biological functions are done by proteins. Okay, 98%. 1 second. Sorry, 98. What is happening? Yeah, 98% of all biological functions are done by proteins. What are like some of the functions we can imagine a protein can do? Our cells are repaired by proteins. Cell division is supported by proteins. Okay, the structural integrity is provided by the proteins. Okay, the signaling is done by the proteins. Then it acts as enzymes. Okay, all the enzymes are proteins. Okay, so if you keep on counting and counting, antibodies are proteins. So proteins are fundamentally performing most of our biological functions. Okay, so one of the key characteristics of proteins that we can talk about is protein is a very, very complex structure. So if we reduce our protein, let us take one of the best protein that we can imagine. So example is hemoglobin. Guys, please mute your calls. Okay, please mute your calls. Okay, so anyways, let's continue. Yeah, proteins, when we talk about proteins, proteins is a very complex structure. So you know that we have red blood cells, and red blood cells have hemoglobin, which is one of the major proteins essential for carrying oxygen, right? So let us talk about hemoglobins and take it as an example to understand the structure of proteins. So hemoglobin has an iron, okay, a ferroheme group or iron group, okay, or ferric group, which is present in the center. And we have four chains that are attached to hemoglobin. Sorry. It's not working. Okay, give me a second. I'll just try to help you with another presentation. Okay. Guys, please mute your phone. Sorry, please mute your devices. All right. So just give me a second. I'll show you the structure of a protein so that we can understand even better. Okay. Chalo. So here is the protein structure. Okay. Here is the protein structure. So using this protein structure, we can understand, okay, how the protein is made. Fine. So as I said, when we talk about proteins chains of amino acids, and these chains of amino
Lasting a
acids we
capital
call it
W.
as polypeptide chains. Guys, just mute, no? Yeah, this is a participant. Give me a second, people. Yeah. Hello, students. I'm sorry for the disturbance. Let's go ahead. Okay. So I said protein consists of polypeptide chains. Okay. So if you can see here, there is one chain here, there is another chain here, and there is another chain here. So they're all coming together to form a three-dimensional structure, which we can determine as a protein structure. So if I tell what is polypeptide chain, let's go back a little bit. Okay. We'll just go back a little bit. Proteins are made up of polypeptide chains, and polypeptide chains are nothing but polymers of your amino acids. Okay, so it's nothing but polymers of your amino acids. So what are amino acids? So if I take, okay, let me make you understand. We have carbon, okay, we have methane. Okay, what is the structure of methane? CH4, isn't it? So I have carbon which is attached to 4 hydrogen groups. Now what will I do? I will start replacing this one hydrogen atom, and I will start attaching different groups. Okay, I will leave this hydrogen atom alone. So look at this left side, okay, left side hydrogen atom, and look at this group. Okay, in the left side, what did I do? I removed this hydrogen atom and I attached a NH2 group. Okay, this is called as an amine group. Okay, next one. So that is why we get this amino acids. All right. So next I take out this hydrogen group, and then I will add this group. So you can see here COOH, right? So I will replace it by COOH group. And then we have another hydrogen atom. I will replace it with a functional group. Then I have a general structure of an amino acid. So it's very easy. You can just remember like this, right? So an amino acid will have an N terminal, which is what charged amino acid. So we have an N terminal that is a positive charge, and C terminal, which can give us a negative charge. Are you understanding? So this N group and the C group are present in an amino acid. Now, when you have one amino acid and you have another amino acid, they have to attach each other to form a chain, isn't it? So how do they attach themselves? So we have C terminal of one amino acid and N terminal of another amino acid, and they form a covalent bond between them. And this we call it as a peptide bond, which is a covalent bond. So once they form this bond and they form this chain, so this one chain of amino acid is called as polypeptide chain. One polypeptide chain or a group of polypeptide chain, they go under structural modification to give rise to what? Proteins. Did you understand, people? So we have different types of amino acids. Basically, if you see from bacteria to all the organisms, we can come across 20 different types of amino acids. Okay. In the 20 different types of amino acids, 9 are essential amino acids and 11 are non-essential amino acids. Now, what is essential and what is non-essential? Now, if I eat food and if my body depends on a particular amino acid based on what I eat, okay, the only way I can get this amino acid is through my food, then that is called as essential amino acid, which are 9 in number. Non-essential amino acids are the ones our body has the capacity to prepare on their own. Are you understanding? These are called as non-essential amino acids. So some of the examples of the amino acid and their structure is given here. You can see very clearly. We have an N terminal, we have a C terminal, and then we have a hydrogen group. Look at the functional group here. If there is H, we call it as glycine. Are you understanding? So similarly, if we have another group, okay, we have another group that is called as CH3 group. If we have a CH3 group, that is called as alanine. If we have CH2 group, see here, CH2OH group, then we call it as serine. So based on what R group they have, the amino acids change their characteristics. Okay, we have positively charged amino acids, we have negatively charged amino acids, we have neutral amino acids, we have polar amino acids, we have non-polar amino acids. Okay, so I'll share a chart with you. So it usually when it comes to second year, we'll talk about the characteristics of amino acids in detail. So that will help you to understand different types of amino acids. Clear? So what did I tell you? Amino acids will help us in construction of polypeptide chains. Polypeptide chains undergo structural modifications to give us proteins. Clear? So you can see here, glycine, lysine, valine, glutamic acid are some of the examples of your, see, if you take acidic amino acids, we have glutamic acid, we have basic amino acid, we can take it as lysine, we have neutral amino acids, which we can consider it as valine. Understood, people? So they all change their characteristics based on their functional group, and which is very, very essential in determining the characteristic of an amino acid. Understood? So examples for essential amino acids are lysine, leucine, isoleucine, and tryptophan. So these are all the names of amino acids, people. Similarly, non-essential amino acid, we have examples like glycine, alanine, serine, and arginine are some of the examples for non-essential amino acids. Clear? So fine. Amino acids, they exhibit one of the important characteristics. Okay. So if I put an amino acid, okay, in a solution, okay, what kind of a solution? I will just put them, in a water containing solution. Okay. So if I put them, they will act as a Zwitter ion. What is a Zwitter ion? So they will start exhibiting two important charges in the same structure. In the N group, it will have a positive charge. You can see here on the right end. And in the C group, they will start showing the negative charge. So the structure having both positive and the negative charge, okay, this is called as a Zwitter ion characteristics of an amino acid. Okay, so fine. Did you understand? So let us go for lipids. Guys, are you understanding? I'm sorry for the troubles with respect to PPTs. Okay. So next onwards, we can go, easily and make it more simpler. So now we have to talk about lipids. So what are lipids? Lipids are basically water insoluble substances, so which are containing carbon, hydrogen, and oxygen, and they are long chains. Okay, lipids are actually long chain fatty acids. So they're usually water insoluble. So lipids are of several categories. We have simple lipids, we have compound lipids, and we have derived lipids. So let us understand what are simple lipids. So understanding simple lipids comes with understanding fats or fatty acids. Okay. So if I tell something called as fatty acids or fats, okay, fats and oils, you can just take it as an example. In general terms, we call it as fats and oils. In chemical terms, we call it as fatty acids. Okay. So these fats, what we call, they contain two important components. One is a fatty acid component, other one is an alcohol component. Okay. The fatty acid component contains a long chain of carbon and hydrogen. Okay. So these chains can range from almost 8 to 32 repetitions of carbon. So usually in most of the living organisms, they are around 16 to 18 carbon atom repetitions will be there. So this fatty acid chain, it is attached to an alcohol group called as glycerol. Okay, something like glycerol. So take an example here. So when we talk about the structure of your fats, we have an alcohol group. In the left side, we have an alcohol group called as glycerol. And this glycerol contains 3 carbon atoms, carbon 1, carbon 2, and then we have carbon 3. And they have in total 5 hydrogen atoms and 3 OH groups. Understood? So this structure is called as a glycerol. Okay. And then come back here. Look at this long chain. This long chain is made up of repetition of carbon and hydrogen group. For example, if you change the next slide, you can see here. Can you see here the palmitic acid? You can see here there is carbon, 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 and we have hydrogen, 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 hydrogen. So this is called as palmitic acid, which has a COOH group at the end. So this COOH group will give its acidic nature. All right. Now what will happen to this glycerol? Okay. Now a fatty acid chain will come attached to this glycerol. If one fatty acid chain attached to one glycerol, this is called as monoglycerides. If two fatty acid chain attached to one glycerol, it is called as diglycerols. If one glycerol has 3 fatty acid chain, it contains triglycerols, okay, or triglycerides. Mostly or the things that we consume fall under the category of triglycerides. Is it understood, people? So very easy to understand. Fats and oils, they are made up of two important components. One is an alcohol group called as a glycerol, and second one is the fatty acid chain. So they attach, okay, to the glycerol. If one is attached, it is monoglyceride. Two, it is attached, it is diglycerides, and three, it is attached, it is called as triglycerides, and that will give us fats and oils. Understood? So based on this, we can divide this fatty acids into two important types. One is saturated fatty acids, other one is unsaturated fatty acids. So when it comes to saturated fatty acids, okay, so you can see that you don't have any double bonds or triple bonds. Okay, if I take carbon, 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 and carbon, and it goes on, you can only see the single bonds. If I have only the single bonds, okay, then it is called as saturated fatty acids. Why do I call it as saturated fatty acids? Is because I can no more add any element. Okay, I cannot add any element or an atom into this structure. It is saturated. It does not share any bonds with any of the elements. So example is palmitic acid, which is a saturated fatty acids. Usually the saturated fatty acids, what they do is they become hard in the cold climates. Okay, so saturated fatty acids are known to cause several problems to human beings, right? So they usually clog our blood vessels, okay, and give us the risk of heart attacks and blockage in our hearts. When it comes to unsaturated fatty acids, we have two different types of unsaturated fatty acids. And these two different types of unsaturated fatty acids are cis fatty acids and trans fatty acids. So I told you the unsaturated fatty acids do not have any double bonds, whereas unsaturated fatty acids do have double bonds. So let us take an example. If I take an example of a carbon chain, C, 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 okay? So let us say we have a hydrogen here and we have a hydrogen here. All right, people. So if there is a double bond here and if there is a double bond here, so they're present on the opposite side, isn't it? They're called as trans unsaturated fatty acids. Okay. If it is present on the same side, it is called as cis unsaturated fatty acids. So examples are oleic acids and arachidonic acid. Usually cis fatty acids are very good, which we get it from cod liver oil, or we get it from olive oil. Okay, they're very good for our health. They're known to boost our immunity, and they're also very, very important for our cell repair and the constant construction of our plasma membrane. Whereas trans fats are human made. What do we do for trans fats? We take saturated fatty acids and we pump hydrogen. When we pump hydrogen, they are known to form this alternate double bonds called as trans fatty acids. So usually when you eat the snacks, you can see on your labels, there is trans fat. So trans fats are very, very bad for your health. Okay. So that is about the types of fatty acids. Clear, people? Now, when we talk about compound lipids, so in compound lipids, we have one of the important lipids called as phospholipids. Now, if I ask you, what is the composition of your plasma membrane? Immediately, you're going to tell that it is made up of lipid bilayer, correct, right? Your plasma membrane is made up of lipid bilayer. That's what you're going to tell. So, what kind of lipid is present in our plasma membrane? Our plasma membrane is made up of phospholipids. So why did all the living organisms choose phospholipids? They chose it because our phospholipids have one of the important characteristics, such as they behave like amphibians. Okay, what is an amphibian? They live on both water and they live on land also, right? So similarly, phospholipids also act like amphibians. That means they're both water loving and water hating at the same time. How is it even possible? Okay, let us take an example. If I give you a structure of a phospholipid, now look at this lecithin, which is a phospholipid. Now you can come to two important points. See, can you see here? This is the glycerol. C is there, no? This C and C is a glycerol. To one of the carbon atom, there is a phosphate group attached. Are you following me, people? So wherever, see, for example, we have C, and C. All right, people. So let us say that we have, for one of the carbon atom, we have a phosphate group attached. Understood? And for another carbon atom, we have one chain of saturated fatty acids. I will write it as saturated fatty acid, and then we have another chain of unsaturated fatty acids. Are you understanding? So it has a head and it has a tail. So the head, that is the phosphate containing head, is water loving. Understood? And then whatever the tail we have, which is made up of fatty acids, they are hydrophobic in nature, that is water repelling or water hating in nature. So this is called as hydrophobic end. So since they have hydrophilic and hydrophobic end, these phospholipids are called as amphipathic molecules, right? Amphi means amphibian. Amphipathic means showing the characteristics of both water loving and water hating at the same time. So take an example, people. You know what scientists did? Scientists took these phospholipids and they dropped it in water. When they dropped it in water, these phospholipids, they arranged themselves in a very unique fashion. How did they arrange? Let us say that we put a drop of phospholipid in the water. When we put it in the water, all these phosphate heads, they were pointing outward, that is towards the water, and all these tails, right? All the tails of unsaturated and saturated fatty acids, they were pointing towards the inside. So what can you understand by this? Outside, what is there? Water is there. Inside, what is there? There is no water. So these tails are water fearing. That's why they organize themselves towards the inside. Whereas the phosphate groups are water loving. So they're okay with interacting with water. So they organize themselves to the outer surface. Understood? So this is a very important characteristic in determining the lipid bilayer characteristics of our plasma membrane. Correct, no people? Right? So in our plasma membrane, we have two layers, isn't it? You can see this. All these heads of the plasma membrane are what? They are water loving. Sorry. They are what? Water loving. You can see that all the insides of the plasma membrane. Just look at this right diagram. You can see like tail-like portion where I'm pointing at. All these tails are water hating. Since our living system consists of majority water, that's why our plasma membrane to the outside of the cell and towards the inside of the cell has to be made up of water loving compounds or phosphate heads. And between these water loving compounds, we have water hating region, which is made up of fatty acids. Okay. So that is why phospholipids are the best examples for compound lipids, and they're the major constituents of our plasma membrane. Did you understand, people? Are you following it up? Yes or no? Students, can you give me confirmation? We have 1 minute. Okay. Chalo. So students, please come on time to the class. I understand there might be some technicalities, some technical issues. Okay. So definitely I'll make sure that this chapter is continued and revised again, because it's a very, very important chapter for both +1 and +2. All right. So we'll come back again and we'll approach this chapter and we'll try to finish this chapter, completely. So you have a better understanding. Clear? Is it fine, people? Today we had a lot of disturbances. I'm sorry for that. Okay. So, okay, people, thank you very much. I'm going to end the session, and we're going to have a better session next class. Okay. Thank you very much. And I'm going to see you in the next class. Bye-bye.